Hello and welcome to the lecture series on basic cognitive processes. I am Dr. Ark Verma from IIT Kanpur. Today we will be talking about the physiology of visual perception. Now you might wonder in the course on perception, why am I limiting this to visual perception? But visual perception per se is the most important, or probably it's better to say the most investigated form of perception. Obviously you have all the senses, you have the sight, you have hearing, you have olfaction, that is smells, you have gustatory, that is taste, you have tactile, that is related to touch. All of these senses basically contribute to this perception, but the most investigated area, or something that has been most studied in cognitive psychology, is this area of visual perception. Okay, the eyes, or say for example, the most important information that probably we get out of our surroundings is through the eyes, and in that sense, that kind of skewness in the amount of research that is done in each of these areas. Also, traditionally in courses in cognitive psychology, visual perception is generally something which is given slightly more importance. I'm not saying that I'm following the convention here, but we'll begin with understanding more details about visual perception in the end, and if time permits, we might talk about auditory perception and related processes as well. That said, we will talk about the basic part, or the first entrance into what visual perception as a process is like. So, how does this really begin? We'll try and understand the physiology of visual perception. Function, or say for example, how do our eyes see? What really helps our eyes see, and what are the very basic processes? I'm not going to go into a lot of detail about each of these. The idea is just to give you a flavor of what are the constituent processes in visual perception. Okay, so let us begin. The beginning point, or the initiating point of visual perception, is basically the registering of light in your eyes. Okay, so the precondition for the fact that you can see anything, for that matter, is the presence of light. If there is light, you see. If there is no light, you don't. Okay, so it's kind of a sort of a binary there. Now, what is light? Light is basically electromagnetic radiation that can be physically described in terms of wavelength. There is this range of wavelength which we are more sensitive to, and that basically forms what is called visible light for us. Okay, humans can perceive only a very limited range of this wavelength of this electromagnetic radiations, and this visible wavelength basically falls between 380 to 750 nanometers. Sorry. Here you can see this range of visible light. So there is this electromagnetic radiation, and right from 380 nanometers to 750 nanometers is what we can actually see. So any electromagnetic radiation, or the electromagnetic radiation in this wavelength, is what constitutes our visible spectrum. Now, how does the process of vision really begin? Where does this, how does light contribute to us seeing? So, vision basically, or our, sensation of anything in this world through the eyes, start when light passes through this protective covering of the eye, the top part of the eye, which is the cornea, which is a clear dome that protects the eye. We'll talk about that. I can actually show you the figure and come back here. This is what the eye actually looks like. You can see there are these different parts here. You can see that there is a pupil, there is cornea, there is a part called iris, there is a posterior chamber wherein the lens is situated, and then there is this ciliary body which has those muscles. The eye is filled with some substance which is called the vitreous humor. At the back you can see there is things like the retina, there is the fovea, and there is an exit nerve which is called the optic nerve. So again, I'm just taking names of these different areas. We'll talk about them in much more detail as we go ahead. Now, what is so you can see what is the cornea. Cornea is basically a clear dome that is protecting the eye, something that is the outer layer of the eye. Then through this cornea the light passes through the pupil. Pupil is basically the opening in the center of the iris. You can again refer to the figure. Here you can see that there is this pupil which is the opening in that thing called the iris. You can see that already in the figure. Just a moment. Yeah. So light kind of so it starts through the cornea, passes through the pupil, which is basically the opening in the center of the iris, and then it continues through this crystalline lens which you saw, and the vitreous humor, which is a gel like substance which is filled in the eye, and then it reaches at the back. So you see the eye starts at the cornea, goes through the pupil, passes through the lens, passes this entire gel like vitreous humor, and goes at the back. At the back is where the real processing of the light starts. So the light then focuses on what is called the retina. You might again refer to the figure. At the back there is this retina. Retina is basically where the electromagnetic energy, that is light, is transduced, that is converted into neural electrochemical impulses. If you are interested in what transduction is, any signal from the external world, you might remember in the psychophysics class we've talked about it, any signal from the external world, any sensation needs to be converted into a signal that can be understood by the brain. So the electromagnetic radiation, which is light, needs to be converted into neural impulses so that the eyes or the brain, more importantly, can make sense of it. So the light passes through the cornea, goes through the pupil, passes the lens, and the vitreous humor goes and falls back at the retina. At the retina is where this is converted into electrical impulses. That is where the brain starts making sense of this. The vision is most acute at the fovea. The fovea is a very small thin region of the retina, which is basically at the size of a pin. Okay, just like the size of the head of the pin. Okay, the retina basically contains layers of neuronal tissue. These neuronal tissue basically includes different kinds of cells, cells like the ganglion cells, the amacrine cells, horizontal cells, bipolar cells, and then also photoreceptor cells called the rods and the cones. We will so here you can see a more clear picture of the cornea, the iris. You see the pupil is there, there's the retina at the back, and you'll see that bulge just like a pin, the head of the pin. That is what the fovea is. Here there is this layering of these different kind of cells. So the first layer of the neuronal tissue at the retina is closest to the front outwards facing surface of the eye. So the first layer is the layer which the light falls first, so outwards facing surface of the eye. This first layer is basically the layer of what is called the ganglion cells. Okay, the axons of these ganglion cells, if you remember the chapter on perception, the axons are actually the part which kind of protrudes from the cell body. The axons of these cells constitute combine together to form what is called the optic nerve. So this is the outer facing surface of the cell, the ganglion cells which form the optic nerve. I'll show the figure to you now. So you can see that there are these ganglion cells in the layer number 3, the axons of which are forming what is called the optic nerve. Okay, coming back. The second layer consists of 3 kinds of interneuron cells. These cells are the amacrine and the horizontal cells and the bipolar cells. The amacrine cells basically make single lateral connections to adjacent areas of the retina in the middle layer of the cells. The bipolar cells make dual connections. They make connections forward and outward to the ganglion cells, as well as backward and inward to the third layer of these retinal cells. So you'll see these connections. Now you can see the bipolar cells being connected to the inward rods and cones, and also to the ganglion cells. So they are basically connected to both inside and the outside part. The third layer is basically the layer which consists of what are called photoreceptors. As you can guess from the name, these are the cells which basically convert light energy into electrical energy that is transmitted by the neurons to the brains. So this is where exactly this process of transduction is happening. There are 2 kinds of photoreceptor cells, rods and cones. Each eye consists about 120 million rods and around 8 million cones. Both of these cells have their own specialities. I'll talk about them in just a moment. Now, rods and cones simply do not only differ in shape, but they also differ in their compositions, what they're made of, their locations, and how they respond to light. Before we move further, it might be helpful to see these cells again. So you see that there are three layers. The first layer, or actually the third, the layer number 3 is basically your ganglion cells. The layer number 2 is your bipolar cells connected both inwards and outwards, and the layer number 1 is basically your photoreceptors. You can see the rods and cones in different shapes there. Okay, so you can see how light is really moving. Light is energy is entering the eye and it's triggering photochemical reactions in rods and cones at the back of the retina. Chemical reaction in turn activates the bipolar cells. From the bipolar cells, information is going to the ganglion, and from there it is going to your optic nerve, and via the optic nerve it actually transmits to the brain. Now, what are these rods and cones doing? These rods and cones basically have substances which are called photopigments. Photopigments are chemical substances that react to light and transform physical electromagnetic energy, which is light, into an electrochemical neural impulse that can be understood by the brain. Now, electrochemical neural impulse, we've talked about this in the physiology part, where we were talking about neurons, action potentials, and those kind of things. The rods are long and thin photoreceptors, more highly concentrated in the periphery of the retina than in the foveal region. So, rods, you will find they are more concentrated in the outside regions of the retina, and they are basically responsible for night vision and are sensitive to light and dark stimuli. So, rods are basically which are more useful when there is less light in night kind of scenarios, because they are basically reacting in binary to whether there is light or whether there is darkness or absence of light. The cones are very short, they are slightly thicker photoreceptors, and they allow for the perception of colors. Okay, they are highly concentrated in the foveal region than in the periphery, because this is where the light is focused more. This is where the acuity of understanding of really seeing shapes, really seeing colors is best. So the rods are more sensitive to black and white, the colors are more sensitive to different, and the cones are more sensitive to different kinds of colors. Here you can see the difference basically in rods and cones. You will see they are not only structurally different, and they are also functionally different, as we discussed already. Now, the rods, cones, and the photopigments could actually not do their job if they were not really connected somehow to the brain. So we have to talk about how all of this really connects to the brain as well. So the neurochemical messages that are generated in this section of the eye, produced by the rods and the cones, travel via these bipolar cells to the ganglion cells. The axons of the ganglion cells in the eyes collectively form what is called the optic nerve for that particular eye. You can see that in the figure that we saw earlier. So this is how this communication is actually happening. The optic nerve basically of the two eyes join at the base of the brain to form something called the optic chiasm. The optic chiasm basically, or at this point, what happens is that the ganglion cells from the inward or the nasal part of the retina cross through the optic chiasm and extend to the opposite hemisphere of the brain. So for example, I can show this to you through this figure. You can see this figure here. You will see that there is an inward or nasal part, and there is an outward which is basically the retinal part. Okay, so the inward part basically is where this crossover is happening, and input from one eye is going to another hemisphere. Input from say for example, input from the left eye is going to the right hemisphere. Input from the right eye is going to the left hemisphere. This crossover point is called the optic chiasm. Just going back, so at this point, at the optic chiasm, the ganglion cells from the inward or the nasal part of the retina cross over through the optic chiasm and extend to the opposite hemispheres of the brain. The ganglion cells from the outward or the temporal area, so the inward area is called the nasal area, the outward area is called the temporal area, which is actually near the temples. The temporal area of the retina goes to the hemisphere on the same side of the brain. So this is how this crossover happens. The lens of each eye basically inverts the image. So you might have done elementary physics in your class 6th or 7th. You might have known that a particular lens kind of forms an inverted image of the world as it projects this image, and the same happens with the lens of the eye, so it projects an inverted image to the retina. After being routed through the optic chiasm, about 90% of the ganglion cells then go to what is called the LGN, or the lateral geniculate nucleus. This is a set of nuclei in the thalamus. Okay, so thalamus is the basic region of the brain wherein this is going to. From the thalamus, what happens is that the neurons carry information to the primary visual cortex, which is the first area in the occipital cortex that kind of receives this information, the primary visual cortex in the occipital lobe of the brain. And this occipital cortex basically is where the detailed processing of this information coming from these, from the retina starts to happen. Okay, so this visual cortex or your occipital lobe, it contains several processing areas. Each of these areas really carry out different kinds of analysis on the incoming visual information. Okay, and these areas can differ into how they process the intensity of light, how they process the quality, how they process the color, the depth information, the location of something, the kind of pattern there is, the form there is. See, visual perception is a really complex activity. I can take an example. I can say that if I'm looking towards you, I'm actually looking at multiple levels of information. I'm looking at colors, I'm looking at contours that are shapes, I'm looking at whether it is a two-dimensional or a three-dimensional image, so I'm looking at depth as well. I'm making sense of how far you are from me, that is distance. So I'm doing, and I'm also, say for example, processing whether you're moving or not. Isn't it? So even though visual perception, typically might seem rather simple, but there are so many processes happening at the same time with anything you're looking at. So if you look out and you are kind of able to figure out that there is a car moving on the road, you are actually doing a lot of these analysis over there. You have the basic shape of the car, you have the colors, you have the information that it is moving, you might get to know that what kind of car it is, so you're kind of doing that kind of analysis as well. So doing these multiple levels of analysis on whatever visual information is coming to your eyes via light. Let us have a look at this figure. This is how this is really arranged. So you have a binocular field wherein both eyes are getting the input, but you also have the right visual field and the left visual field. Okay, so right visual field is predominantly basically what the right eye is looking at, left visual field is predominantly what the left eye is looking at. You can see here in this figure that part of the information from each of the visual field goes directly to the contralateral hemisphere. Contralateral is basically the other side of the optic chiasm. So you can see in this figure the information in the right visual field goes first to the left visual cortex. The information in the left visual field goes first to the right visual cortex. I am saying first because generally these contralateral connections of these ganglion cells are found to be faster than the ipsilateral, basically the same side connections. So this kind of also leads to really interesting or important effects. We'll talk about them in due course when we are actually talking about more things about visual perception. For reference, here is the figure which presents what is called the visual cortices. You can see that there is this lateral geniculate nucleus which is connected to the brain. You can see light is entering through the eyes. It is kind of through the optic nerve reaching this area called the lateral geniculate nucleus. That is what your thalamus is about, and from there the information is being relayed to what is called the occipital lobe, which is the lobe which has to do with vision. And you can see these so many areas here. Okay, each of these areas are referred to as V1 to V8. You can see, and you can see what the specializations of each of these areas are. Say for example, I'm just reading out for you. Areas from V1 basically is called the striate cortex. It catalogs whatever input is coming in. Area V2 basically is more responsible for relaying the signal to higher association areas. V3 basically processes form. V3A processes motion. So these are both the areas are in the same place. Then you have areas like V5, V6, V7, V8, those kind of things. Basically, so this is how the processing of information starting from light till coming to the brain really happens. This is basically all about the physiology that we will be learning in this particular lecture. We will go on to how these different areas in the occipital lobes perform visual processing in the coming lectures. We'll talk about form perception, depth perception, consonancies, those many different things, and how basically you make sense of a particular visual stimulus, all of that in the coming lectures on perception. Thank you.